Hi, my name is Rachel Dumais. I'm a second year graduate student in the Genetics and Molecular Biology PhD training program here at UNC Chapel Hill. I grew up in Front Royal, a small rural town in Virginia. I was recruited to swim at the University of Richmond, and that is where I had the opportunity to find my passion for scientific research. Before I got to college, I didn't have a clear concept of what it meant to do science or how you become a scientist. It wasn't until I stayed at Richmond over the summer to train for swimming that I saw my peers applying all the things we had learned in class to their summer research projects. And that made me think that research was something that I could do and possibly even enjoy. I joined the developmental and evolutionary biology lab in the summer after my second year at Richmond. It was an incredible experience, and I've been pursuing a career in science ever since. After I graduated, I worked in Richmond for 2 years, and then I started my graduate work at UNC in the fall of 2019. I joined Rob Dillon's lab, where I currently study the gut microbiome and how it affects fat metabolism and aging. A microbiome refers to the genetic material of all the microbes that live in and on an animal. According to data published by the Human Microbiome Project, the microorganisms associated with the human body outnumber human cells 10 to 1. I'm particularly interested in the bacteria that make up the gut microbiome because the biodiversity of the gut microbiome has been associated with a lot of aspects of human health, including drug metabolism, disease progression, obesity, cancer prognosis, and brain function. Therefore, one of the keys for living a long, healthy life may be probiotics. Probiotics are live microorganisms, such as bacteria, that are intended to have health benefits when consumed or applied to the body. Many people in the US already take probiotics as part of their daily routine. However, the probiotic market is not regulated for quality control by the FDA, meaning no clinical trials or approval process is necessary to confirm the safety or effectiveness of these products before they go into the market. If probiotics are going to become a routine part of preventative care, rigorous and reproducible studies on the bacteria marketed as beneficial for human health are needed. Because the architecture of the human gut is very complicated and many of the bacteria present in the human gut microbiome are unknown, I am using C. elegans as a model system to explore how bacteria in the gut of an animal influence fat homeostasis and aging. C. elegans are a free-living nematode worm species found all over the world, and since the 1960s, it has become one of the most abundantly used animal model systems. C. elegans are great for studying bacteria-host interaction because their guts are composed of 20 identical epithelial cells and can be colonized by one or many bacterial species depending on the food source provided. My hypothesis is that there are some compounds made by the bacteria in the gut of an animal that interacts with host molecular machinery to influence gene expression and ultimately alters the way the animal processes and stores fat. If you're interested in learning more about this project, have questions about how to get involved in research at UNC, or want to know more about my journey to become a scientist, please feel free to reach out.